Okay, it's my pleasure to introduce Ximena. She's a biology student, a PhD student at the University of New Mexico, specializing in fish immunology. Her research in the Salinas lab at the Center for Evolutionary and Theoretical Immunology is in collaboration with the research team from Israel, where they are investigating and characterizing the effects of a novel nidovirus recently discovered in rainbow trout farms in northern Israel. Thanks so much for being here today.
Thank you for the introduction. So, as you mentioned, yeah, today we are going to talk about a new discovered virus, the trout granulomatous virus, and its impact in the gills of infected rainbow trout. So, this is the content we are going to talk today, and let's start. So, in the left side hand, you have this QR code. You can scan it, and it's going to redirect you to the journal where the paper is published, and you can know more details about the discovery of this new virus. Because now I'm going to give you like a short introduction or short background. So, in the 2018, several rainbow trout farms in northern Israel started to experience some outbreaks of an unknown pathogen. And the main feature seemed to be the enlarged livers with nodular masses in their parenchyma. Although the prevalence, as you can see here, 4 to 7%, it is impacting large fish in the fattening stages, which might indicate that it's a chronic disease. So, about the key characteristics of the TGV virus, sorry. Although the researchers tried to grow the virus in different rainbow trout cell lines, so far those efforts have not been successful. So they instead use electron microscopy as well as metagenomic sequencing to reconstruct the whole viral genome. And they concluded that it belongs to the order nidovirus, and because of the feature of the liver, they decided to call it trout granulomatous virus. So let's talk about the core findings of this TGV infection. Interestingly and relevant with what I'm going to tell you later, the first signal of disease that was noticed in the diseased fish was that the gills become really pale, suggesting anemic in a chronic disease. Also, unpublished data suggests that the gills might be the first entry point for the virus. Additionally, the fish weren't eating or also had stunted growth, were less responsive to external stimuli. And important to highlight is all of these non-pathognomonic symptoms cause economic losses, even when the disease is not leading to mortality. And as I mentioned before, the main post-mortem feature was the granulomas in the liver, but also it was reported damage in the heart and in affected fishes, and also distension of the coelomic cavity, as you can see here in the pictures, and ascites compared to a healthy trout. So with this background, we thought the gills might play an important role in the pathophysiology of the virus. So we aimed to characterize and evaluate the damage in the gills and its immune response against the TGV virus, as well as to guide vaccine development with our immunological findings. So we received different paraffin blocks from 8 rainbow trout fish coming from infected farms in Israel, and in red are highlighted the fish that has the lowest city value, meaning that those fish have the highest viral load. And because of the aims of this study is the gills, we are going to focus on these gill samples. So the first thing we did based on this high viral load in the gills was to develop a pathology score system with 6 different features, as you can see here. And we scored the overall damage between 0 and 5. And interestingly, as you can see here, the fishes with the highest viral load, the 3 and the 6, have also a huge overall damage. But interestingly, fish number 5, that doesn't have the high viral load, as well has overall damage. So in here, you can see that the pathology scores did not correlate with the viral load. And I'm going to show you some examples of what we saw with the histopathology findings. So as you can see here, we saw a lamella fusion in the black arrows are signaling. We also saw clots, hemorrhage or blood clots, as well as we saw this infiltration of immune cells, specifically eosinophilic granular cells or EGCs. We also saw here you can see edema or epithelial lifting, where the secondary lamella starts to detach and starts the fusion. And additionally, we were really surprised when we found in this hematoxylin and eosin staining that were granulomas in the gills, not only in the liver, as I mentioned in the background. And also because, as far as we know, it hasn't been described the granulomas in the gills. So it's a really interesting finding. So in here you can see the granulomas in the gills. So here in the right image, we can see here the starting of the formation of a granuloma in the primary lamella, where the secondary lamella was already fused. In here, we also can see two different size granulomas, as well in close to the cartilage, and also in the primary lamella, where the secondary lamella was already fused. And most interestingly is that we found this big granuloma in the interbranchial lymphoid tissue of the gills. So from these pathologies, the next question is what cells in the gills are getting infected? So with the virus. So we developed an antibody against the N protein of the TGV, and we validated in the infected liver, and this is what we got. So we saw, you can see here, red dots. We saw expression in the areas next to the cartilage, where I mentioned to you before that they're the immune infiltration, and that we also see these TGV positive cells within the granulomas. And because of these findings, we wanted to see if the infected cells were immune cells. So we ran a double staining with the TGV N antibody plus MEC class 2 antibody. And indeed, yellow arrows here, you can see there is a sort of colocalization between the TGVN antibody and the MEC class 2 antibody, which could suggest that the virus infect immune cells. We also measured these cells, and the diameter of these positive cells is approx 8 to 9 micrometers. So this guides us to think it could be monocytes or not. And then we also did the single MEC class 2 staining, finding that besides the expression close to the cartilage that I mentioned you before, we also saw here MEC class 2 expression in those cells, these thin and flat cells that looks like endothelial cells. And these also are forming these kind of pockets or pools where the secondary lamella started to fuse. So interestingly, and based on the literature, endothelial system get activated by infection and start to turn on MEC class 2, usually associated with hypertrophy or altered function of the endothelial system, which might be a maladaptive immune response. We also inquired about the IgM expression in those infected fishes, and in here, the arrows, as you can see here, the IgM positive in B cells as we would expect it. But when we saw here the infected fishes, we started to see this different pattern of accumulation of IgM. In these pockets where I mentioned you before, it was this damage in the endothelial system. So we don't know whether this IgM comes from the serum of the fish, and it's accumulated there, of it of if this accumulation are from deaths from cells that used to be there and then died. And the only thing we can say in this moment is that this pattern we can interpret it as a maladaptive immune response. And with those results, we wondered whether those pools or pockets of IgM were accumulating because of the lack of the polyclonal immunoglobulin receptor, or PIGR, which is the molecule to help to transport these secretory immunoglobulins across the epithelium. So perhaps if there is a lack of this PIGR, maybe this is the reason why we saw these pockets of IgM in here. And in fact, when we run this PIGR staining, we saw that there is, in here you can see there is more expression of PIGR compared to the healthy trout. So with these results, we do not think that the IgM pockets that we saw in the past slide are accumulating because of the lack of PIGR. Although this pattern expression also could be a maladaptive immune response, but at least we can say that, as I mentioned to you before, the accumulation of IgM is not because of the lack of this molecule. So in conclusion, what I've told you is that the TGV, which is a new viral disease, causes granulomas in the gill during high viral infection. Also infects immune cells, which allow us to think that it could be a mechanism for hiding from the immune system, and in that way facilitate its spreading across other organs. We also saw that causes endothelial system damage and immune activation. And finally, as I mentioned to you at the beginning of the talk, gills might be the first place of infection. Therefore, mucosal vaccination should target this tissue. Special knowledge to Salinas Laboratory, our collaborators in Israel, and our funding. Thank you. I'm happy to receive your questions.
Great presentation, Ximena. Folks, if you've got questions, feel free to unmute yourself, or you can drop them into the chat. Well, it's a quiet group today. Thank you very much. You did a great job.
Thank you.